Okay, so we've looked at graded potentials. We know that they happen because we have chemical gates that open. We know that they're short-lived, that they decrease in their strength, and that they can add up. So we call them graded potentials. Action potentials or nerve impulses are the major way excitable membranes of neurons and muscles communicate. They do not decrease in strength. They occur in milliseconds and they only occur in axons, and it's not a chemical gate that causes them to open or happen. It's a voltage gated channel. So let's look at the steps of an action potential. First, you have a polarized or resting membrane. All voltage gates are closed. The membrane is what on the inside? Negative on the inside, positive on the outside. Because of a graded potential that sent a wave of depolarization down the membrane, depolarization happens when sodium activations open for 1 millisecond and allow sodium to rush in. The membrane is now less negative, or what we call positive, on the inside. Notice that the gate has two channels. Now we've opened one gate and we're very quickly going to close it with the second gate. So it's got two gates on it. So now we are positive on the inside, negative on the outside. As soon as the sodium gate closes, the potassium slow channel opens and potassium rushes out. So the sodium inactivation gate closes, potassium gates open. Remember, there's more potassium on the inside of the cell than the out, so potassium rushes out to reestablish membrane potential, or negative on the inside and positive on the outside. So we have repolarized the membrane, and now it's ready to receive another signal. Potassium channels are a little bit slower to close. They're slow channels, so it allows too much potassium to leave out of the cell, so the inside of the cell becomes more negative than it should be at rest. We call that after shoot or after hyperpolarization, and so then the sodium potassium pump is going to be working and bring us back up to our negative 70. So potassium channels are slow, so too much comes in and the potential is a little too negative. This is hyperpolarization or after undershoot, but it is the sodium potassium pump that will restore it. Remember, we have 3 sodiums that are pulled out and 2 potassiums are pulled in. So first we are polarized, negative on the inside, positive on the outside. We open up sodium channels. Sodium rushes in, causing it to be positive on the inside, negative on the outside. The sodium fast channels close, potassium slow channels open, potassium rushes out and reestablishes negative on the inside and positive on the outside. Now, action potentials, or because they are voltage gates, can propagate down the entire length of the axon if threshold is reached. So if threshold is reached, with an action potential, the impulse becomes self-propagating or self-generating. It continues all the way down the axon. So we have that depolarization here, which causes more sodiums to move, which causes depolarization there, and sodium moves and causes depolarization there. So it moves all the way down the axon, and it continues at a constant velocity, and the membrane ahead of it is depolarized, the membrane behind it is repolarized. It's kind of like a domino effect. Now, I think probably a better analogy would be the keyboard of a piano. If you push the first key down, you could say that that was your action potential, and you move from the first key to the second key to the third key to the fourth key all the way down the keyboard. Well, what happens to the key that you take your finger off of? It pops back up. So where your finger is the wave of depolarization, and what happens afterwards? Repolarization. Action potentials are all or none. You reach threshold and they fire, or you don't reach threshold and they don't fire. It's just like if I had a gun, I can pull real hard on the trigger, it'll still fire, or I can pull soft on the trigger, it'll still fire, or if I don't pull enough on the trigger, it won't fire. They happen if threshold, a negative 55 millivolts, is reached. A sub-threshold, which would be too little of a stimulus, won't cause a response, and a super-threshold stimulus will cause the same response as a threshold. It's all or none. The stimulus strength can then change the same way we change the muscle strength, either by the frequency of the stimulus or let's do frequency of the stimulus first. So the stimulus strength is coded by the frequency of impulses. And I think I gave an example in the muscles, like if you had a headache. If you had a mild headache, it might be an action potential being sent like that. But if you have a really bad throbbing headache, they may be sent like that. So that stimulus strength is a lot more than one that's like that. When sodium channels are open, the neuron is unable to receive another impulse. This is during the absolute refractory period. But during the relative refractory period, when potassium channels are still open, the neuron will respond to a suprathreshold stimulus. So let's look at this graph of the activity in a neuron. Right here is when the stimulus is hitting the neuron. We open up sodium channels, which cause depolarization, and then sodium channels close, potassium channels open, and we start repolarization. During this period of time, it doesn't matter what you hit that neuron with, it will not respond. But once we get down into the relative refractory period in this area of depolarization, if I hit it with a stronger than negative 55 stimulus, it will jump up and fire again. And so it'll fire with a greater response. So conduction or velocity, or how fast an action potential, is based on the axon size. The larger the axon, the faster the impulse will travel. If it has myelin sheaths, that's going to insulate it. And so a myelin sheath, the conduction of the impulse is just going to be at the nodes of RANVA. And so that's going to be a lot faster than if it has to be continuous down the whole axon. So if there weren't any myelin sheaths, we have to travel every inch of that road, and we're going to be a lot slower than if we just have to travel the nodes to get to the end. So continuous is where there's not a myelin sheath and it's slower. Saltatory is where we jump from node of RANVA to node of RANVA, and it is faster. So the sodium channels are concentrated at the nodes, and so that's where you're just having the depolarization wave. It's 30 times faster with saltatory conduction. So an unmyelinated axon, the voltage-gated sodium and potassium channels regenerate the action potential at each point along the axon, so the voltage does not decay. Conduction is slow because movements of the ions and of the gate channels take time and must occur before voltage regeneration occurs. With a myelinated axon, myelin keeps the current in the axon. It doesn't decay, so action potentials are generated only at the nodes and appears to jump rapidly from node to node. Now we also put nerve fibers into 3 groups based on whether they're big and fast or slow and small. Group A are the large myelinated fibers, and they actually can send impulses at a rate of 150 meters per second. And you're probably saying, well, how fast is that? That's about 300 miles per hour. That's pretty darn fast if you think about sending a message from my toe to my brain. And you know, I'm only about 1.5 meters tall, so think how fast it goes. Group B, oh, I'm sorry, and that would be like somatic sensory to the skeletal muscle, motor of the skin and skeletal muscles and joints. Group B are intermediate in size and they're lightly myelinated, and they're going to send an impulse 15 meters per second, which would be about 30 miles per hour. That's a lot slower, but still, think if you're only 1.5 meters high, that means that you know how fast that is from one side to the other. And those are like autonomic nervous sensations like pain and temperature, some of your mechanoreceptors and flex responses. The smallest neurons that are unmyelinated only have a velocity of 1 meter per second. But again, I'm only about 1.5 meters, so even on my smallest unmyelinated fibers, I can send an impulse from my toe to my brain in about 1.5 seconds. That's still pretty fast. 2 miles an hour. And that would be like pain and temperature and itch, some of the automatic nerve reflexes. Now, I found this joke when I was looking up information about these groups of neurons. Let's see if you get it. The guy is bragging about his car, and he said, "Buckle up. We're going to go as fast as an action potential and an A beta nerve fiber." And she says, "Oh, really? I doubt if this old thing can go as fast as a C nerve fiber." So he thinks he can go 300. She thinks he can go about 2 miles an hour. So what happens when the action potential reaches the end of the axon? Well, remember when the nerve impulse ended the end of the axon terminal, we had a neuromuscular junction. We remember that calcium came into the neuron, the axon terminal end bulb, and caused it to release its vesicles of neurotransmitters into the synaptic cleft. Let's see how that differs when we're connecting nerve to nerve. It's still going to be a synapse because it's between two excitable cells. We can have a synapse between an axon and a dendrite, which is the kind we see most often. With an axiodendritic synapse, the presynaptic neuron conducts the impulse towards the synapse, and the postsynaptic neuron transmits the impulse away from the synapse.
Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse? Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse? Neurons communicate across junctions.
I guess my movie is not going to play all the way through, but they were talking about chemical synapses, and that would be a synapse like between a muscle and a nerve, where you send a neurotransmitter across the synapse. We also have other kinds of connections between neurons. We have an axon that will synapse with the soma. We can have an axon that will synapse with another axon. We can have a dendrite that synapses with another dendrite, or a dendrite that synapses with another cell body or soma. These are less understood and less common, so scientists are not real sure how they function or why they're different from the others. So that was a chemical synapse. Now let's look at an electrical synapse. Electrical synapse is where you have gap junctions, where you have connexons that connect one cell to another. And what that provides for is the message going to a whole group of cells so they can respond together. So it synchronizes activities. We find them in the brain and in embryonic tissue to get embryonic tissues growing at the same pace. We can also find electrical synapses in the heart, so that the electrical signal that controls the pacing of the heart goes through a whole area of an atria at one time. An example is in the hippocampus of the brain. They synchronize emotions and memories so that you have the appropriate emotional response to a memory. You don't want to think about the death of a loved one and start laughing hysterically. So they synchronize so that those match up. Gap junctions or electrical synapses. So let's go back to chemical synapses because these are more understood and easier to see. And it's kind of like we talked about with a neuromuscular junction. So this is the axon terminal bud. What we see is when the action potential gets to the bulb, it opens up calcium channels and calcium flows in. Calcium flowing in causes the vesicles to merge with the membrane and release the neurotransmitters into the cleft. The neurotransmitters go across the synaptic cleft and bind to ion channels on the other side. So it's a chemical that crosses the synapse and opens up the channel. Well, one difference is that with the neuromuscular junction, the only neurotransmitter we had was acetylcholine, but with neurons synapsing with other neurons, we have a whole bunch of neurotransmitters we can release. Some of them will actually go across and open up channels to excite the next membrane. Others will go across the channel and inhibit the impulse from going any further. Some of them will make that membrane more negative and further away from threshold. So let's look at the notes. The flow of information or ions between excitable cells is by way of neurotransmitters that open and close ion channels. There is a slight synaptic delay as the chemical crosses. So here I think is the rest of my movie.
There is a narrow gap, the synaptic cleft, between the synaptic terminal of ascending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
So let's put those steps into the notes. The impulse reaches the axon terminal of the presynaptic neuron. So the impulse, the action potential gets down here. What kind of channels open? Calcium channels open, and calcium rushes into the axon terminal. When it rushes in, it causes synaptic what? What are these bubbles called? Synaptic vesicles to release their substance into the synaptic cleft. What is that substance that's going to continue the communication? Neurotransmitters into the space between excitable cells. This space between excitable cells is called the synapse or the synaptic cleft. Neurotransmitters diffuse across the cleft and bind to the postsynaptic membrane. The postsynaptic membrane ion channels open to excite, or they can inhibit to bring it further away from threshold. If an ion channel opens and excites the next membrane, then it's an excitatory postsynaptic potential, or if it brings it further away from the threshold or stops, it's an inhibitory postsynaptic potential. This is a graded potential. It's going to be short-lived. It's going to die out. It's usually found in dendrites and somas. Neurotransmitters degrade by either diffusing away or being absorbed or being broken down by enzymes. So here is one showing this one is an excitatory or excitation postsynaptic potential where the stimulus causes it to go towards threshold. This is an inhibitory postsynaptic potential where it causes to go further away from threshold. So neurotransmitters degrade by enzymes from diffusing away from the synapse or by astrocytes absorbing them. Remember, an astrocyte is a neuroglial cell found in the central nervous system. So what kind of modifications can we have of the synaptic events we've just talked about? Well, one I've been kind of hinting about is called summation. And if you remember in the muscle what summation was, it's where we kind of add together several different potentials, and if they reach threshold it can turn into an action potential. It would be like if you had a 70 and a 90 and a 40 and an 80 and a 100 and they all average together, and if it's passing, then you pass the class. So a graded potential can add together to influence the postsynaptic neuron because the axon helix acts as a bean counter until threshold is reached. You can have temporal summation where one neuron sends impulses at increased frequency. So if I hit this dendrite from this neuron over and over and over and again, like 10 and 10 and 10 and 10 and 10 and 10 and 10 until it got to 60 and you passed, you had the action potential. It's all at the same point. That's temporal. Or it could be spatial summation where you're going to have several different dendrites receiving a message and they're all adding up to be. So like if this one was 40 and this one was 20 and it added up to 60 and it fired the potential, or negative 55 if you will. So if it's in the same place with increased frequency, it's temporal, or if it's in several different of the processes going into the soma that all add up to potential, it'll fire it. Another modification is known as synaptic potentiation. Repeated or continuous use of the synapse exercises the nerve and enhances communication and postsynaptic response. That's why if you study material and you quiz yourself every day and the information builds and builds and builds, those synapses become stronger so that when you try to recall it on the test, it comes up a lot quicker than if you looked over it one time. Another type of modification is the presynaptic inhibition. The release of the neurotransmitter is released. So something causes, when that action potential gets down here and that calcium comes in, that is stopped. We're not going to send the neurotransmitter across. Some painkillers do that. Now, how about those neurotransmitters? With the neuromuscular junction, we saw one, acetylcholine, but with nerves to nerves, there are more than 50. They're classified by their chemical structure and by their function. I'm not going to require you to learn all the different types, but you can see acetylcholine is one of those, but if you look in the charts in the book, there are more than 50. Now, your brain is very complex and there are organized neural pathways all in your brain, and it's very complicated to see all of these pathways. Let me see if I can get this YouTube video to play.
Billions of brain cells are not done forging links with billions of other brain cells. Eventually, there will be trillions and trillions of connections between cells. Eventually, there will be trillions and trillions of connections between cells, charged with electrical pulses, rippling like lightning storms across the hills and valleys of the brain's deeply furrowed tissue. Every cell in its place, every link between cells carefully organized. Nothing random, nothing arbitrary.
What we would really love to understand is how the brain during development generates millions and millions of neurons, sends them to the right position in the brain, and then somehow instructs each of those individual nerve cells to form very, very specific connections with one another. You can think about development like a play that follows a script that is written down by the genetic code, but it has no director, it has no producer, and it has a bunch of actors that have never spoken their lines before. Despite all this, you pull the play off. To me, that's a miracle.
So as you see from our YouTube video, the brain is very complex and there are millions of pathways, and all those neurons are in the right place, hooked to the right one, sending the right message. So when we look at integration of the neurons in the brain, there are a few things that we need to understand. Neurons function in hierarchical groups, and these groups must be fused or integrated together. So scientists know this. Neurons in the central nervous system are organized into groups called neural pools, and they're connected by functional circuits. Let's look at the different kinds of circuits. One kind of circuit is a diverging circuit, where you have an impulse coming into one neuron and it splits that impulse and excites 2 neurons, and then 4 neurons, and then 8 neurons. So some kind of action where you're wanting it amplified would be a diverging pathway. One brain neuron innervates hundreds of skeletal muscles. Another kind of circuit we see is a converging one. And if you look at this one over here, we have 3 different neurons all sending an impulse or an action potential to this neuron, who then fires and sends it out. So you're sending a lot stronger, so to speak, signal. Converging circuits, different sensory neurons or impulses can elicit the same memory. Like I can smell an apple pie and think of my grandmother's house, or I can see a picture of my grandmother's house and think of my grandmother's house, or I can hear my grandmother's voice on a tape and think of my grandmother's house. So 3 different senses, and I'm all thinking about or having the same memory of something that happened at my grandmother's house. Another kind of circuit is a reverberating or oscillating circuit. These kinds of circuits are like the circuit that keeps you awake. So it's going to keep firing over and over and over to keep you awake and alert. Another kind of circuit is a parallel aftercharge, and an example of that would be mental processes like math, where you have input coming in and you diverge along several pathways. You're thinking, oh, how can I solve this problem? And several different pathways come back together and you get the answer. An input can be processed either in a serial pathway, like a monosynaptic arc, reflex arc. So a stimulus is sent down a sensory neuron. It synapses with an inner neuron. We figure out what to do. Synapses with a motor neuron, and we send a message to the effector, and that causes us to have a sensation or a pain from that nail in our skin. That's monosynaptic. Or it can be a parallel circuit, where you can have several pathways going. You send an impulse along the sensory neuron. It goes into the spinal cord and actually has 2 different diverging pathways. One is going to go to the muscle so that you have a response of contraction of this muscle, and then you're going to have another pathway being sent to this muscle to relax, and you're probably going to have a third pathway that goes up to the brain so you know what's happening. So we can either run it serially along one pathway, or we can split off and run a couple of pathways. So That really was the chapter on nerves and nervous tissue. Now, if you'll see on your note sheet, kind of the rest of the chapters I've put all into facts about the central nervous system that you're required to know. In lab, you dissected the brain, and so you know that there are four parts, major parts to the brain. They are the cerebrum, the cerebellum, the brainstem, and the diencephalon. The cerebrum is sometimes called the executive suite. The cerebellum is responsible for motor, command center, equilibrium, and balance. The brain center is a survival center with a lot of the automatic processes that occur. The diencephalon includes the thalamus, which is the gateway to the cerebrum, and the hypothalamus, where some of your visceral autonomic commands are located. It's an area that maintains homeostasis. There are 5 cerebral lobes. They're associated with the bones that cover the cranium. 4 of them are. The frontal bone would be here, so this is the frontal lobe. Parietal, occipital, and temporal. Now, if you pull the temporal and the frontal apart, down deep you'll see the deep lobe of the insula. So what are the functions of each one of those lobes? The frontal lobe is responsible for reasoning, intelligence, motor, and speech. Parietal lobes are primary sensory. Occipital is for visual. Temporal is for auditory, and the insula is for taste. What are the 3 regions of the brainstem? It's going to be the medulla oblongata, the pons varioli, and the midbrain. And this whole brainstem is the survival center. So the medulla, what it does, is a connection between the higher brain and the spinal cord. There are centers there for heart rate, respiratory rate, vomiting, coughing, sneezing. The pons, or pons varioli, is a conduction pathway between the higher and lower brain and between the cerebrum and cerebellum. There are some areas there that help control respiratory rate. And then the midbrain is a conduction pathway between the higher and lower brain. This is where you find the corporal quadrigemina with the superior and inferior colliculi, and that's where you have visual and auditory centers. Now, your brain is surrounded by the cranial cavity. It's in the cranium, so the bones help protect the brain, but you have some other ways to protect the brain and spinal cord. There are 3 membranes that surround the central nervous system, and they are called meninges. The outermost meninge is the toughest one. It's very thick in comparison to the other ones. It's called the dura mater, which is Latin, stands for tough mother. Then you have where the blood vessels are associated with, and so it looks kind of like a spider web. It's the arachnoid mater, or the spider mother. And then the layer that is deepest that adheres to the brain tissue is the delicate mother, or the pia mater. Going between these meninges is going to be cerebral spinal fluid, which also cushions and nourishes and takes away waste products from the brain. It helps to form the brain-blood barrier. So when you look at the cerebrum, the surface of the brain, you can see hills and valleys, or grooves and ridges. So the grooves are called sulci, or a sulcus. The ridges are called gyri, or a gyrus. I kind of think if you're down in the groove or down in the valley, you're sulking, so it's sulci, so the other one must be gyri. Now there is a deep fissure that separates the 2 cerebral hemispheres, and it is called the longitudinal fissure, and we'd be looking at it right here. Now, I said the cerebral spinal fluid also helped to protect the brain. In your brain, you have some cavities called ventricles. You should have learned about this in lab. You have the 3rd ventricle that's up above the thalamus, and then you have the 4th ventricle that's underneath the cerebellum, and then on either side of the pellucidum that septum that separates the two hemispheres or holds the two hemispheres together, is the lateral ventricles. And so it's in these ventricles that you have choroid plexuses, masses of blood vessels, and the nutrients and oxygen that is in the blood is filtered out and becomes cerebral spinal fluid. So the choroid plexuses of the brain ventricles produce cerebral spinal fluid. So just some other little facts about the brain. What is the limbic system? It is the cerebrum and diencephalon region where we find emotions and feelings. What is reticular formation in the brain? It's in the brain stem, and it keeps the cerebral cortex alert and conscious. How about cerebellar peduncles? That was going to be in the cerebellum for balance and equilibrium. And the hippocampus, that's part of the limbic system that gives you long-term memory. Now, just as a review, Chapter 11 has many new terms. You should be able to define words like polarization, depolarization, repolarization, synapse, diverging circuit, converging circuit, neuroglial, and each type in the central and peripheral nervous system. What is a tract? What is a nerve? What are the nodes of Ranvier? What's a stimulus? What's the nucleus? Or a nucleus? What is a ganglion? What is serial processing? What is an IPSP? What is an EPSP? What are the divisions of the nervous system? What is saltatory conduction, and how does it compare to continuous conduction? What is a myelin sheath? What is absolute and relative refractory periods? So just to remind you of a little bit of this information, let me show you a few movies again.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential. Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary. Generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts. Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
And that ends the notes on the nervous system. I was talking to the thing.